Many important functions of our bodies depend on proteins. They account for more than 50% of the dry mass of most cells. In fact, their importance is underscored by their name, which comes from the Greek word proteus, meaning primary. Each protein is made up of amino acids which are covalently bonded to each other by dehydration reaction on ribosomes within cells. These linkages are called peptide bonds, and the amino acid chains are called peptides. But not all peptides can be called proteins. The difference between these two terms is that a protein is a biologically functional molecule. However, the function of a specific protein is determined by the unique characteristics of amino acids from which it is composed. All amino acids share the same structure. They are organic molecules with both amino and carboxyl groups. This means that an amino acid can act both as a base and as an acid based on the conditions where we put it. Let's say that this molecule is now in an acidic environment. Then the carboxyl group of this amino acid will capture the hydrogen ion, and the whole charge of the molecule will be positive. In this environment, in an acidic environment, amino acids will act as a base. In a basic environment, the amino group will give its hydrogen by forming a water molecule, and the whole charge of the molecule will be negative because of the carboxyl group. So we can say that in a basic environment, amino acid acts as an acid.
You probably have noticed that amino acids also have an R group. An R group is a rest group that is also referred to as side chain, and it determines the unique characteristics of every amino acid. Now let's take a look at the side chains of different amino acids. The side chain of an amino acid can be nonpolar, polar, and charged. Nonpolar side chains are hydrophobic and, as you can see, don't have any functional groups. Instead, they are mostly open hydrocarbon chains like glycine, alanine, valine, proline, leucine, and isoleucine. These are also called aliphatic side chains. There are also aromatic side chains in a nonpolar group, like tryptophan or phenylalanine. Now, polar side chains, in contrast, have functional groups, like hydroxyl group in serine, threonine, and tyrosine, sulfhydryl group in cysteine, or carboxamide group in asparagine and glutamine. These functional groups also make them hydrophilic. And finally, charged side chains can be charged positively, like lysine and arginine, or negatively, like aspartic acid and glutamic acid. The properties of the side chains of amino acids in the protein determine its unique shape and structure. Usually, amino acids with hydrophobic side chains tend to stay inside the molecule, away from the surrounding water. All proteins have one of four existing structures. Primary structure is a simple sequence of amino acids, whereas the secondary structure occurs only when the amino acid sequence starts to fold into more complex structures, like an alpha helix or a beta-pleated sheet. Tertiary structure is when a protein folds into a three-dimensional structure, and the quaternary structure is a structure of a protein that is composed of multiple amino acid sequences as subunits. Thanks for watching this video, and I'll see you in the next one.